Yeah, hello. My name is Joe Fenster, and if you watch this video, you want to know how immune protection against corona actually works. Yeah, this video should explain how immune protection against corona works. In other words, how does a natural infection protect against the next infection? How does a vaccine protect against an infection? And of course, as the coronavirus evolves, how can an immune response against a vaccine or a natural virus help to protect against the next variant? And how does this actually work? To understand all this, you need to know basically three things. You need to know how antibody-based protection works. You need to know how this is induced, the immunity against coronavirus, from a natural infection and from the vaccine. And third, you need to know how the kinetics is of the immune response. And this knowledge is the basis of decisions like, should we vaccinate against a future variant with an original vaccine, new vaccine, should we vaccinate some age groups at all in the future, and so on. Okay, so let's start with antibody-based protection. So this video should exemplify how antibody-based protection against COVID-19 works. So this is first of all telling you how do the initial steps of the SARS-CoV-2 infection pathway look like, how the protection against this virus happens, and what's the problem if you have variants, and finally, what isotypes are involved in protection. Okay, so let's look at the first steps of infection. Of course, you already know that the virus acts primarily in the respiratory system, and infection is via the respiratory system. So we have to look there how the infection works, and the cell layer which is involved is this respiratory epithelial cell layer. So what happens if the virus gets to this layer? You see that there are receptors on the surface, and the virus simply adheres to these receptors. And if we look more closely, we see that the SARS-CoV-2 virus is composed of different things. And on the surface, it has the so-called spike protein. And this spike protein interacts with the ACE2 receptor on the surface of this epithelial cell. The names are not so relevant, but it's a specific interaction of the surface protein of the SARS-CoV-2 virus and the protein which is there on these epithelial cells. And as soon as you have this interaction, this virus is taken up by the cell. And as soon as it's inside, of course, it's hidden from anything which are antibodies and so on. So in other words, if you want to protect against the virus, what you have to do is you have to prevent this initial interaction. Now, how can you do that? Well, the secrets are antibodies. Now, let's look at the first scenarios. We have a scenario. I just drawn a part of the viral particles here, just the relevant one, which is the surface. And let's have a scenario where we have antibodies against this spike protein, which are present in a sufficient amount. So what happens is simply that these antibodies bind to the spike protein, and if they have bound to the spike protein, the virus cannot bind anymore to the ACE2 receptor. Now, this is one scenario. What happens when the antibody levels go down? Now, if the antibody levels go down, it's quite straightforward. The problem is you don't have enough antibodies anymore to bind to all surface proteins of the virus. And in this case, of course, the level of antibody protection is not sufficient because you still can have free spike proteins which can bind receptors. So if you want to have a full protection against the infection, these initial attachment steps, this is what you want to protect, then you need to have a high level or a high enough level of antibodies which are specific against your spike protein. Now, what happens if we look at this interaction and the spike protein more closely? You might realize that we have a protein which is optimal against our antibody, but which is not perfect against the receptor. So if I mutate my spike protein and I optimize this interaction, and this is what the virus consecutively does, then my antibody protection will not be optimal anymore. So I will have a low binding affinity to the antibody and a high affinity to the receptor. Initially, I had a high affinity to the antibody and a low affinity to the receptor, a relatively low affinity to the receptor. So the evolving viruses, new variants, will get higher and higher affinity against the receptor and a lower affinity against prior existing antibodies. Now, what happens if a virus infects us which has a mutated spike protein, what we have not seen before? So, for example, we have vaccinated against the original variant, and now we have a mutant variant, despite the fact that we have enough antibody levels. So if in such a scenario, I will have enough antibodies, however, the binding of the antibodies is not optimal anymore. So in this video, you see that they don't bind as fixed as before. So despite the fact that I have enough antibodies, unfortunately, I have not the full protection, because these antibodies kind of can flip off this spike protein again. So this is one thing what we have to take into account if we look at antibody-based protection. But the other thing we need to take into account are isotypes which are required for protection. As we talk about the epithelial cell layer, respiratory epithelial cell layer, not all antibodies are there. So let's say the normal antibodies what we induce are the antibodies in circulation, and you see they're in circulation. So these antibodies cannot go out and be on the surface of these epithelial cells in our system. And you see that there are special antibodies which can go through this layer and then mediate this protection. And this is a so-called IgA isotype, and the antibodies which are in the circulation are the IgG isotype. So for protection, I need this IgA isotype, and I need high enough levels in order to be able to protect. So the previous video has shown how antibodies can protect against SARS-CoV-2 infection. This video shows how immunity against SARS-CoV-2 actually is induced. So how antibodies are induced, first of all, via a natural infection or vaccination, how an antibody response can be induced against a mutated virus, and how a process called affinity maturation helps in fitting these antibodies to the new viral spike protein, how killer cells are induced, and a comparison of different immunities induced by natural infection and vaccines. So in the first video, we have shown how the virus attach to the cell and how antibodies can block this. So obviously, as soon as the virus is in the cell, antibodies are of no use, and the virus can start the viral replication. So how does this work actually? So in the case of the SARS-CoV-2 virus, it's taken up, and then the virus gets out of this lysosome. Of course, this is simplified in this step, and then we have an RNA which is in the cytoplasm, and this RNA. Then with this RNA, I have a replication of the virus, so all the viral proteins and RNA are being produced. This is very simplified here, but the cool thing is to understand the immunity, it's not so important that every detail is correct in this case. The basics are, as soon as you're in the cell, the virus starts with producing viral particles and the viral proteins, of course. And one protein which is being produced of the RNA is again the spike protein. Now, this spike protein, if it gets out of the cell, either in a full viral particle or as the protein itself, it can get into contact with a cell called a naive B cell. And you see on the surface of this B cell, you have a receptor which resembles an antibody, and you will see later on it's nothing than a membrane-bound antibody. And as soon as this antibody, which fits to the spike protein, this B cell receptor, identifies the spike protein, the B cell gets activated, and then it starts to proliferate. So you simply get more B cell. By the way, during this step, it loses this expression of this B cell receptor. And as soon as you have enough cells, then soluble antibodies are being secreted. And these are the antibodies we have seen in the other video, which can actually protect against the infection because they can block this binding of the SARS-CoV-2 spike protein to the receptor, the ACE2 receptor. And these cells which produce that are so-called plasma cells, and these are then, of course, the cells producing this anti-spike antibodies. The origin was the viral RNA, but at the end, it doesn't matter whether the spike protein is produced by the viral RNA, so whether a viral replication happens there. You just need this spike protein. And this is the basic principle of the vaccines. So a vaccine which is a protein vaccine, you simply use a protein which you inject, and you have directly the spike protein in your body. Now, these RNA-based vaccines are in principle mimicking the viral RNA and then also produce in the cell the spike protein. And a DNA-based vaccine, first of all, has to produce the RNA. Of course, there's some more technology behind these vaccines. So you need to protect the RNA. You need to be efficient in taking it up. Then there are some modifications both in the RNA and in the spike protein to make it stable. But these are details. These are not so important. The essential of the process is that the vaccines do more or less the same as the virus. However, of course, an RNA-based vaccine, for example, just produces the spike protein, so therefore no virulent particles are being produced. And second, of course, it's an RNA-based vaccine, so therefore risks like integration or so in DNA in the chromosome are not there because it's RNA. Now, if I have vaccinated, or if I had a natural infection and the virus is eliminated, of course, for vaccination, it's not eliminated, it's just the response, the initial response against the vaccine is over, then there is something staying in your body, and these are soluble antibodies, which I didn't have before. Now I have soluble antibodies in a high level circulating against the SARS-CoV-2 virus. And I have so-called memory B cells. So some of these plasma cells stay alive and are acting as memory cells. So this is what I have. And the soluble antibodies we have seen before are the one which can immediately protect against a new infection. An important thing is, of course, the kinetics, because you have a decay of these. So later on in the kinetics section, we'll talk about that, what happens if the antibody levels go down. If I now have an infection with a mutated spike protein, we've already seen that this mutated spike protein, the problem is that the antibodies I have now are no more high affinity, so they loosely attach only to this to the spike protein, and therefore they can only partially protect. What I need is a high affinity antibody which exactly fits this mutated spike protein. Now, there are two things happening if I have an infection with a mutated spike protein, a so-called secondary response. Usually also such a mutated spike protein will be recognized by the B cells I already have, as long as it's not completely different. But usually, at least in the case of viruses like coronaviruses, this is the case. This is usually not the case if I have influenza viruses, but this is a completely different mechanism how the virus evolves each year. Now, in the secondary response, I then produce, of course, again, just antibodies which are specific against this normal spike protein, but I have way more. So if I have more, I might also be able to protect. And therefore, even if the connection is not so high and if it's again going off, then the next thing we see is another antibody. So it might at least partially help to protect against the virus. Now, optimal would be, of course, if I have a directly fitting virus. And some of these memory B cells, by the way, this also happens in natural infection, some of these memory B cells then start a process called affinity maturation. And what you have seen is that during this process, the antibody gets a structure which then fits exactly to this mutated spike protein. Of course, the details how this works are more complicated, but at the end, what happens is that you will get antibodies which fit exactly to the new profile. And then, of course, I get again a proliferation, I have new plasma cells, and then I have the production of soluble high affinity anti-spike antibodies. And these are, of course, fully protective then for the new virus. What I also induce are so-called so-called killer cells. And this is important if I compare then now immunities induced by different means, especially if I have now a new spike protein, which is completely different. Again, this is unlikely that I have a spike protein which is not at all recognized by any B cell. Still, this might happen. And these killer cells are also important for a full elimination because, of course, I also want to eliminate cells of the body which are infected with the virus and produce viral particles. I want to get rid of them. And this is simply done by these killer cells. And how are they induced? Now, we have already said that the spike protein is produced by the viral RNA or by the vaccine RNA. And in this case, this parts of this spike protein can get degraded by a particle which is the so-called proteasome. So this proteasome partially degrades proteins, for example, the viral spike protein, but it could also partially degrade proteins like the capsid protein. So in natural infection, of course, I also have a capsid protein, not in the vaccine, but in a natural infection. So I will get of the viral proteins then a mixture of degraded, partially degraded protein particles. These are so-called peptides in this case. And you see that in the example, I look at one of these parts. You see that if I look at the original spike protein, this comes from a region which is not affected at all in binding. So this is a kind of so-called conserved region in this case. So I would assume that I have the same effect of this partially degraded spike protein, whether I'm infected with the natural or the initial variant or a mutated variant. Now, what happens now with this particle? This particle is first loaded on a molecule which is called MHC1. The name is not so important in this context, and this then presents this molecule on the surface. And now I have a cell population which is called a killer cell. And these killer cells can specifically, again, recognize the spike protein. And as soon as this cell is activated, it destroys the cell which has this spike protein. So in other words, this cell destroys cells which actively produce virus particles. Of course, this cell is also there's an induction, so it's also a bit simplified here, but this is the job of a killer cell. And of course, also here I can form memory and so on. We will see that in a second. Now, if we now compare the immune responses and the memories of different ways of the induction, you see that I can induce, of course, an immune memory via a natural infection, via a vaccine. And now let's compare RNA vaccines, same as for DNA vaccine, and protein vaccines. So in all cases, I will induce antibodies. So vaccines are optimized for the induction of antibodies. So usually I can induce way higher levels of antibodies with vaccines compared to a natural infection. Now, what I also induce are, of course, B cells, which stay there after the infection, these memory B cells. And this is, of course, a bit lower. Let's go down. And you could expect that the level of these B cells is a bit higher after vaccination compared to a natural infection. At the end, we have seen that we can also induce cytotoxic T cells, which kill infected cells. So this is induced more or less, I would say, to the same amount of vaccines, RNA-based vaccines and viruses. This cannot be induced by protein vaccines because the protein vaccines, remember, are outside of the cells. But to be able to induce the cytotoxic T cell response, you need to be inside of the cell. Again, this is kind of simplified, but more or less, this is for sure valid in this case. And finally, I can also induce anti-capsid cytotoxic T cell responses, which is another viral protein. And this, of course, only occurs after a natural infection, as the vaccines do not have the capsids. So we have seen now how these antibodies are induced, how the antibodies act. However, unfortunately, this antibody response doesn't last forever. So to be able to judge about vaccination compared with natural infection and epidemiology, we should know more about the kinetics of the immunity. And that's what we have a look at in the next section. So again, we have already said that the point is we need to induce the B cells which are specific against this spike protein. The problem is that I have only a low frequency of this B cell. So in other words, the majority of B cells I have in my body are recognizing different things. It's just clear because I don't have just the coronavirus. The coronavirus is one virus. So let's assume I just have one B cell in my body which recognizes the coronavirus. Of course, that's simplified, but doesn't matter. I start with just one B cell instead of a couple of hundred. So the first thing I need to do after I have an activation, I need to produce more of these B cells. So, and after this so-called clonal expansion, I have a high frequency of these B cells. Why is this clonal expansion? Because the B cells which are proliferating are just the specific B cells. All the others don't proliferate. And then we know that these B cells can differentiate, and then I have the production of antibodies which protect against this infection. The same is true, by the way, for my killer cells. Also here I have a low frequency and I have an activation to get a high frequency, and then I have the differentiation and I have these killer cells which can kill other cells. That sounds clear, but the question is, how long does this take to happen? We need for a protection these anti-spike proteins, the anti-spike antibodies, or the memory B cells, or the memory cytotoxic T cells. So let's look first at these antibodies and concentrate on these antibodies and B cells. So if I've never been infected before with the virus, or if I have not yet been vaccinated, then of course the immune response needs to mount this antibody level. And how does it actually work? So let's say this is then the so-called primary infection or the vaccination. So this is the first time I get in contact with this virus. I have extremely low levels of these B cells. I said I have just one B cell there, and then I have an infection. The B cell, you remember, gets activated. There's a clonal expansion. I have then a lot of B cells, and then the level goes down again. And I have at the end, you see, I have some B cells left, and these are the memory B cells. So after the infection is cleared, after the virus is gone, this goes back to zero. And the profile looks exactly the same if I have a vaccination with an RNA vaccine, for example, encoding for the spike protein. So my antibody levels, of course, they're a bit delayed because at the beginning, these B cells needs to proliferate first. Yeah, it takes at least 1 week until you start to see antibodies which are specific against the virus. And high levels you have only after 2 weeks. So it's a delay in between until you have the antibodies. If you have the antibodies already there, everything is fine. And you see that, of course, after the infection, you also have a decline of this antibody levels. The cool thing is that the body does not eliminate them readily, so that you drop down to zero, like for the B cells, which is not zero, which is higher, but which is low. You have substantial levels, but of course also these levels decline. So this is of course a time scale which is not necessarily realistic. It's just showing you the overall kinetics here. So indeed, you yourself don't see anything about this antibody production. This kind of happens behind the scenes. What you experience if you have an infection, you have the symptoms. And of course, if you look at the symptoms, you will have full symptoms until this antibody level starts to rise, and then it can go down because these antibodies can act. Of course, in parallel, you have the same with cytotoxic T cell responses, but again, we want to concentrate on antibodies. Now, what happens if this first infection or this first vaccination, there usually, of course, the symptoms are quite low because the symptoms are the activity of the immune system is there. Of course, some people then develop fever and so on. This is normal. However, this is also just based on the immune system, and it's, of course, not so severe as for the infection. Otherwise, an immunization would make no sense. So, but what happens if I wait now for some period? So the infection is over. So you see that there is a decline of the antibodies on the long range. So we have already seen in the first video where we have talked about how antibodies actually protect. it's clear that I need to have sufficient levels against the virus in order to be protective, because I want to fully block the infection. So if I have a level which is above this threshold level, so I cannot specify this threshold level if I have no experience with the virus, but if I have if I'm above a certain level, I have a full protection, and if I'm lower than this threshold level, I only have a partial protection. And what does that actually mean? If I want to have full protection, again, I have to have an antibody level above this threshold, but I also need to have a virus variant which is similar to the initial virus I've been infected or I have been vaccinated. If this is the case, so if I have a vaccine which is active against the same virus which is actually circulating, or when I have been infected already with the virus and I have I'm getting exposed to the virus a second time, what I have then is a sterile protection if I'm above this antibody threshold level. So sterile protection means that I completely block the infection of cells. Therefore, in this case, I will have no viral replication. I will have no substantial symptoms at all, and this is important, I cannot infect other persons because I completely block this infection. And this is important if we discuss about the epidemiology and so on in another video, by the way. So if I don't have now this sterile protection, of course, and because of the low amount of antibodies or lower amount of antibodies I have, I will go through an infection. So I will, of course, then mount a full immune response. So I will have the same as before. I will have my B cells, which start then from this memory B cell level. And then I have the production again of antibodies, which are specific against this spike protein. And as this is an infection which is active, I would also expect in this case to see symptoms. So the symptoms might be less severe, but they will still be there because I'm partially protected, because some antibodies are there. It's better than nothing, but it's not full. If I now would simply overlay the two things that I have now, these are the original kinetics and original levels. What you see is that the rise of these antibodies, of this monoclonal, of this antibodies and B cell is delayed in the case of the initial response. And so therefore it will take longer until I get the protection. And also it takes longer until I can get down with my symptoms. So if I have a first infection or a vaccination, even if my levels are not high enough, I have an initial partial protection due to the antibody levels which are still present. Here, there are no antibody levels if I have not been vaccinated before or didn't have the immunization before. This will reduce my my symptoms level. It's not just antibodies, I have already talked about that, but we can exemplify that very well here. But for full protection, I need to rise higher levels of these antibodies and also my cytotoxic T cells, by the way, and then I have an elimination. But here in an initial primary infection, nothing happens until I have a level which is high enough of these antibodies. So this takes 9 to 12 days until I get a clear clearance of the infection. It's way faster for people who had an initial infection or have been vaccinated, even if the antibody levels are lower. So what is important is I'm lower here, but of course people who have problems, this might be too high still. So we will discuss about the symptoms in a second. If I have said that I have just a couple of memory cells, and it looks like, well, yeah, it's like a friend tells me I had started with €1 on my bank account, now I have €100. And another friend tells me I started with €10,000 and I have €1 million. Then you would say, the 1 million friend is cooler because that's a real difference. This is €990,000 difference. The other is €99. This is, you go 3 times to eat something and then the money is gone with almost €1 million. you have some space left. However, for the immune system, it's exactly the same, because the difference is in both cases 100-fold. And this is exponential growth. It's not additive, it's multiplicative. So you have at the end, to go there, you need round about 6 to 7 doublings. And a B cell needs, it's a fast-growing cell for a eukaryotic cell, but it needs 6 to 8 hours for a doubling. So this means it takes 3 days from 1 to 100 cells. So, and if you have not been exposed to the virus before, for this difference from 1 to 100, it's already a difference of 3 days. And that's a lot, because in these 3 days, nothing happens. 100 cells is 0. They cannot produce enough antibodies to do anything. So you have to wait way longer in order to raise this. This is the problem. Also, intuitively, it looks like, what's the difference? Okay. So, and if I'm now infected with a mutated virus, this is my third scenario, what happens there? Of course, in this case, I know that my antibodies are not fully protective. And if I look at my antibodies, of course, I have the B cells before. You remember that I will have an induction of the wrong antibodies, which happens as before quite fast, and I get high levels. But I need, for a full protection, I need this affinity maturated antibody, and this of course takes a bit longer, but then these will also appear. So also here I expect to have symptoms at the beginning, and also here I expect that it takes some time to clear the symptoms. And if we overlay now the symptoms of the different scenarios, my scenario A is the full sterile scenario. In this case, I have a threshold level of antibodies which is above this limit, so I have a sterile protection. I wouldn't expect any symptoms in this case. Scenario B is the level scenario where I'm infected with but with the identical virus. Scenario C is this level where I have a different virus. So I would expect that again, there is a gradual loss of this productivity due to the levels and the affinity. And it takes longer, but this is if I'm not immunized. And if you compare that, the scenario B and C, to the non-immunized one, I would expect in all scenarios a lower severity of the symptoms. And I would expect that I have a couple of days after the infection so here's the infection. You see that after 3 days, you might start this. Of course, this can differ. So this is a simplified presentation, but you would say in a standard scenario, you would expect to have symptoms after 3 days, which rise dramatically if you're not immunized, which rise to a moderate level if you have the same virus, bit more severe if it's a mutated virus. And then after 3 days, this starts to go down if you have been exposed before. But for the non-immunized, it takes way longer until that goes down. And of course, if you're healthy, young, and so on, you have no problem. You can stand these symptoms here, these severe symptoms. However, if you have problems, it's too high maybe to have the symptoms. However, if you're extremely off risk, maybe even these 3 days of symptoms which are moderate might be too high. In this case, your only option is scenario A. You have to repeat the immunization as soon as this antibody level drops in order to keep this protection sterile, either repeat or an adapted antibody. Now, what happens if I have a variant which is highly mutated? So usually a variant which is completely not cross-reactive with any antibody from the previous infection is unlikely. So it's not just one antibody you induce many antibodies and against the spike protein, many different antibodies. It's unlikely that you don't induce any antibody which actually works. If in the absence of any cross-reactivity, despite the fact that this is unlikely, the more conserved type of the memory will be still there. So I'm talking about this cellular memory. We have seen that this T-cell response is often directed against more conserved epitopes. And this only occurs, this conserved response can only happen if I have RNA-DNA vaccines or previous infection, at least for the cytotoxic T cell response. There are also other T cells, but this is not what we want to talk about here. And then the symptoms, of course, depend on the degree of cross-reactivity and most likely are in between this scenario C and the non-immunized. And of course, a reasonable scenario would be that the initial symptoms are close to a non-protected person, but the severity of the symptoms will decrease more rapidly. So that's it. So this is how this kinetics look like. And of course, I have said that at many places. Of course, both the mechanisms are simplified. And of course, there is a variability in the kinetics of the symptoms. Certainly people who have been immunized and had symptoms for a longer period, and you had no people for a shorter period. So this is, of course, kind of a standard, and there are variations around that. However, it's reasonable. So it's certainly reasonable to state that if you're immunized, even if a virus variant arises, it is likely that you're no more fully protective. That's likely, and you have seen why that happens. But it's likely that the symptoms are lower than without vaccination, and the clearance of the viral infection, and therefore the disappearance of the symptoms is faster. So this is how it works. Of course, it's ideal if you're vaccinated or if you have recovered from an infection, that your antibody levels are high enough in order that you don't get infected anymore. However, if the variants appear, this is the realistic scenario, you will have some infection. However, this infection will be milder, and this is something which is important to conclude with. It is to be expected that viral variants which will come are variants which have a lower virulence and a higher infectivity. And we will talk about that in the final video, where we talk about, so what does that all mean? How a test works, how the vaccine works, and what is it all about at the end? How should we act how should politicians act? How should we act if we have all this knowledge? Now, I want to thank for your attention and looking forward to see you again in the next video when we put everything together. And I wish you that you stay healthy. And if you're infected or have been infected with corona, that you only experience mild symptoms because you have been vaccinated before, and you recover as fast as possible. Goodbye.